Hi, my name is Natalie Leger, and I'm going to be talking about microbial diversity. The extent of microbial diversity continues to elude scientists. The oldest microbes are prokaryotes like bacteria and archaea, and they were the first cellular organisms to ever live on Earth. For some perspective, Earth is 4.6 billion years old. 3.9 billion years ago, bacteria began to evolve and reproduce in Earth's oceans, and 2 billion years ago did microbial eukaryotes evolve. Life on Earth stayed solely microbial until just 0.9 billion years ago. Microbes have had the run of the planet for the longest, which also means they have the most opportunity to evolve. An amazing result of all of this microbial evolution is cyanobacteria. Cyanobacteria evolved 3 billion years ago just before eukaryotes. Cyanobacteria oxygenated the atmosphere and gave rise to microbes and eventually larger organisms that can aerobically respire and use oxygen to make ATP. A good example is Vibrio fischeri, a marine bacterium who exists as a facultative anaerobe. Facultative anaerobes prefer no oxygen, but can sometimes switch their metabolisms to use oxygen if it's present, like Vibrio fischeri. Vibrio fischeri has co-evolved with the Hawaiian bobtail squid, providing bioluminescence and living in its light organisms where there's low oxygen present. The way that we track the evolution that these microbes have been doing is by phylogeny. Phylogeny is the study of the evolutionary history of organisms, and scientists use anatomy, fossils, and DNA to get a look at its ancestry. So by sequencing the genes of 16S rRNA, we can actually tell what the species is of the organism and species that it's related to. RDP, the Ribosomal Database Project, has so far sequenced 3.3 million 16S rRNA sequences as of August 14, 2020. But that may seem like a lot, but it only tells us 17,000 species from all of that data. And scientists have estimated that there's actually 10 million to 1 billion microbial species on Earth. If we've only sequenced 17,000 species, we have a long way to go. To culture is to classify. The only way to obtain the 16S rRNA is to be able to isolate the microbe in a pure culture. Sometimes this isn't even possible. It's estimated that only 0.1% can be grown 0.1% microbes can be grown in laboratories. That means that if there truly are 1 billion microbial species, 0.1% of that, or 10 million species, can be grown in the laboratories and classified. That means out of the 17,000 we've found so far, we still have 983,000 species to go. That means we'll never know 99.9% of our microbial neighbors. They're practically aliens. Our world is teeming with life, both in the marine and the terrestrial environment, and microbes take so many different forms. Who's to say they aren't any more unknown as alien life is? For reference, here's Mars's ocean 3.8 billion years ago. Who's to say that it isn't as foreign as our own ocean? Thank you for watching, and here are my references.